Last, of course, but not least, we have something completely different. We're going to talk about interventions and I believe in model systems and comparative systems to humans. And this will be by Dr. Rafael DeCabo. He is a senior investigator at the National Institute of Aging, chief of the Translational Gerontology branch, and chief of the Experimental Gerontology section in the Aging, Metabolism, and Nutrition unit. So very, very appropriate for him to be here with us. He applies physiologic and tissue-specific molecular approaches to investigate the effects of nutritional interventions on basic mechanisms of aging and age-related diseases. And he is going to talk to us about dietary interventions for healthy aging. He has all the answers.
I wish. Yes. So I want first to thank the organizers for the invitations. And what I'm going to try to do is to give you the perspective from the basic biology of aging, or where we are in the field, and how we are translating some of the findings in the basic biology of aging to humans. So I'm going to start from the very basic and try to work my way up all the way to humans. So one thing that we need to appreciate is that aging is a universal process. It doesn't matter what species you are. You know, these are our favorite type of analysis. You're either dead or alive, and either you're count or not. So there is no, you cannot be half dead, half alive. And what is true is that if you look at the shapes of the curves in every single population, there is like a sigmoidal effect of this. And these are, even if you take mice that they are genetically identical, you will have a very similar pattern of this sigmoidal that illustrates a little bit of how stochastic and how random the process of aging can be. The other thing is, the definition that we go by is that aging are the gradual changes that are in the structure and function of organisms that occur with the passage of time and is not the result of a disease or gross accident. So what are the consequences of growing old? I always use this picture. This is my grandmother, my mom, my sister, and a niece. And this illustrates basically that even when you share environment, culture, recipes, genes, the way that these people are going to walk through the aging process is very, very different. And the time of their life where they're going to die or where they're going to have the onset of the first disease is going to be determined by the interaction between their genes, the environment, the nutrition, and all these changes. What is clear is that as people get old, there is an increase of chances that you're going to have all these appearance of phenotypes and characteristic of diseases of aging. And through the last 15 years or so, we have been identifying some of the key pathways that they have been related to the aging process. We know exactly, or we think that we know exactly, which are the most important pathways that they are altered with the aging process. What is this important? So one of the reasons that is important to understand aging is because the world as a population is getting old. And this is happening very rapidly. So if you look at this graph over here, this is the number of people on Earth under the age of 5, and this is the ones under the age of 65. And this is the projection to 2050. So actually, about now, between right now till middle of 2017, is when, for the first time, the people over 65 is going to be over the people under 5. The other very important thing that I think Roger Fielding said earlier is that the percentage of the portion of the population that is growing more rapidly is actually the ages of 85 plus and 100 plus old. This is the woman that holds the record of the oldest living human. There's 122 years plus a few months. And so far, she was extremely active all the way to like 110. He learned to bike, he is still bike, and he was fencing all the way to up age 100. So what we want is to try to understand and how to intervene to make people to age as healthy as possible. So the other thing that is very important for us is, if you look at the progression of diseases with aging, there is a clear increase, almost exponential increase as you get old. And the major risk factor for each one of these, the 10 top diseases that kill us, is aging itself. It is not obesity, it is not diabetes, it is not cancer, it is basically pure aging. So the bottom line is we're very interested in our mission is to try to increase the quality of human life. We're trying to understand how chronic diseases of the elderly are currently the main limitation of achieving this increase in quality of life. And we're trying to do this is trying to address the major risk factor for all these diseases, which is aging itself. So one question that we will always get asked is, can we really stop this process from going to here to here? And the short answer is no. I mean, there's no way that you can stop it, but in the lab, we've been able to alter both the onset and the progression through interventions, both the onset of disease and the progression of aging. So if you look at the squaring of the human survival curve, you see that in the last 500 years, we have gone from this mean lifespan to this mean lifespan, and the portion of the lifespan that gets increased more rapidly or more dramatically is always a mean lifespan. This is normally, when you square the curve, is a sign of health. And most of this squaring has been done thanks to nutrition, thanks to the healthcare system and public health studies. And the maximum lifespan has not really changed ever since the man is man. Now, in the lab, we can create this shift in the survival curve just by restricting the total caloric intake that animals have on a daily basis. And this is caloric restriction without malnutrition. So these animals are getting all the calories that they need, all the nutrients that they need, but they just have a reduction of the caloric intake. Now, what you see is that there's a shift both in the mean and the maximum lifespan. These black dots represent the onset of spontaneous tumors in these animals, and you can see the black dots are from the control, the animals that they're eating as much as they want, and the restricted animals are the open circle. So you can see that the onset of the first tumor is delayed substantially, and the number of tumors that animals present is also significantly decreased. Another thing that is very important of caloric restriction is not just sheer lifespan. If you look at any organ system, for example, the cancer, even if you induce the cancer and you inject subcutaneously a cancer, you induce carcinogenesis, the animals are protected. And it's not just cancer, it's also if any other organ system that you look at, for example, the muscle. These are muscle from a hind limb of a Fisher 344 rats, age 23 months old, and this is a litter mate that was put on calorie restriction on the same age. So you can see that the structure, the ultrastructure of the muscle gets preserved beautifully throughout the whole aging process, and the function as well is maintained. So we know now that there's a number of species, and this slide is from a paper by Luigi Fontana that published about a year ago, and basically illustrate that almost in every species that we have tested caloric restriction in many different ways, we always have this impact on lifespan and survival. There's always exceptions to this rule, but it's quite a universal and a strong process. So there's always a catch. So what is the catch? That, if you go to a doctor and take you out of calories, that doesn't take that long. And these interventions in mice are long, lifelong interventions. So it's very hard to convince a human to be, for the rest of their life, eating between 30 and 50% less calories every day. Now, how does calorie restriction work? So we know when we decrease energy intake, there's a number of responses that happen from a very immediate time point to takes months to happen. So the understanding of some of these mechanisms in caloric restriction and the aging process have led us to identify molecular targets that now we can use different compounds to actually activate or inactivate some of them. And I'm giving you some examples here, SIRT1, mTOR, AKT, as insulin silencing pathway of AMPK. So we have now some molecular targets that we can actually test whether an intervention that technically is going to be like caloric restriction will have an impact on survival. So the goal was to identify what we call calorie restriction mimetics. So these are compounds that what we're hoping to see is that when you put animals on these mimetics, you will have all the benefits of CR. You'll get to the end of your life completely happy and healthy, and then one day you appear dead. So this is, of course, an ideal situation that normally does not happen, but it is, so like the goal of the research is to try to understand how, with the mechanism that we have learned from studying aging and CR, can we really identify molecules that it will not require the total reduction of caloric intake that you have, but you can actually take them and have some of the beneficial effects. But of course, we've been successful in some of them, but when we went and tested some of these compounds in animal models, the first thing that we noticed was that, rapamycin, that was one of our predictions, had a beautiful effect in survival in males. It was in females, it was a dose response effect, and in the males, for the most part, it responded the same, but there's a substantial difference between males and females. Then acarbose and other of the compounds, worked beautifully in males, but not so much in females. So, we started scratching our heads and say, how is this possible? Caloric restriction always worked. So in the lab, I went back to the drawing board and I started searching through the literature, and there's examples in the literature that says that caloric restriction not always works. So there are strains of mice that caloric restriction doesn't work, some species that don't work. So I wanted to test whether some of these, if these mimetics are not universals, and we know that they depend on a number of factors, can, caloric restriction itself, can we discern or identify ways that you can make a species respond or not respond to it? So what we did is I took two different strains of mice. These are DVA females and C57, one of the most common animals used in rodent research. And this is the strain that is very much used for diabetic research. So what you can see from the get-go, when you put animals either at 20% or 40% caloric restriction, what you can see is, independently of the strain, there is a reduction on body weight. But you can also see that when you have even exactly the same difference, so it's 20 or 40% reduction in caloric intake, the proportion of difference between the al libitum and the caloric restricted one is not 20% and 40%, and it's not equal depending on the strain and the sex of the animal. What was shocking to me was what happened next when we did the survival curves, was that DVA females, it did not really matter if you were at 20% or 40% caloric restriction, you responded identically. So there's no differences in survival. So if you are a DVA female, you definitely do not want to do 40% CR because it's not going to give you an extra benefit. Now, if you are C57 black 6 female, you do not want to do caloric restriction 40% because your life is going to be miserable and you're not going to get any advantage in survival. And the same thing happened more or less with the DVA males. So what this slide illustrates basically is that the response of caloric restriction in this particular case is very much dependent on the sex and the strain of the animal and the proportion of which you are applying the CI, which when you go into trying to translate this into humans, there's a question how we're going to determine if you are a black 6, a DVA, if you're a male, a female. That would be a lot easier. Now, even though we did get this massive difference on response in the strains, the common pathways that are always altered by caloric restriction were altered in all cases, even though if they did not respond to CR, which also raises the question, which of these pathways are truly responsible for either the health or the survival of these particular strains. Now, caloric restriction has moved from our understanding, accepting that implementing 40% caloric restriction in human populations is not going to be possible, to try to see if there are other strategies similar to caloric restriction. For example, time-restricted feeding, where you feed and provide animals food for a very, very short period of time, or if you do intermittent fasting. Sure. If you do intermittent fasting, so you eat one day and not the next, or if you do amino acid restriction, for example, methionine restriction has very similar effect of caloric restriction in terms of survival, or if we use some of these CR mimetics. So in terms, and this is sort of like a global picture to try to understand that basically the way that I like to think about this is the way that an organism senses and processes energy and the energy efficiency. So how once the animal has sensed and ingested the energy is processed through the organism, and how the different steps in metabolism and signaling occur, how these signaling processes can be reduced to very specific biomarkers, so like NAD, ATP, acetyl-CoA, different very, very specific bioenergetic molecules that can directly alter signaling molecules and transcription factors that then drive the changes of what they are called, the biomarkers of aging. So changes in this have been associated with increases both in health and survival. So if you go to the brain, and I promised that I was going to talk a little bit about the effect of nutrition, or at least dietary restriction in the brain, there's been a ton of work done that energy restriction, exercise, and intellectual endeavors cause adaptive responses that they are at the cellular level, and these this is like hormetic stresses. They have produced the reduction and production of enhanced clearance of pathogenic proteins. We know that there is improvement in bioenergetics, improvement in calcium signaling, improvement of the general bioenergetic homeostasis of the cell. And if these two things occur at the same time, then you have an improvement in synaptic plasticity, neuronal survival, and neurogenesis. And that, in the long run, leads to maintaining optimal brain function and resistance to both injury and disease. And I'm going to give you some example from Matt's lab. He's the lab chief of neuroscience in our institute. So for example, in this case, he took mice and he implemented two different types of calorie restriction, either 40% calorie restriction or every other day feeding. What you can see, and also exercise, what you can see here, that wild type animals, of animals that they are prone to be diabetic, if you do calorie restriction and you run, or you do calorie restriction and sedentary, or you do a levitum controlled feeding, running sedentary, you can see a change, a drastic impact on these dendritic spines that they are, markers of memory formation. So you can see that, the sedentary, they have a tremendous effect of diet in the density, spine density. The runners have an improvement from the sedentary to the runner, and then calorie restriction is additive to the role of exercise. Now, if you have just sedentary, you do not have those benefits. And in the case of, and these are control mice, not transgenic for the DBDB, that they don't have diabetes. In the case of the diabetes, they also have exactly the same trend to have beneficial effect, even if you have a very strong genetic drive to have a deregulation of glucose metabolism. There's a number of other papers that Mark has published over his tenure in NIH, and also when he was in the University of Kentucky, that he has clearly illustrated that almost any neurological disorder and any neurological insult can be protected or reversed by the impact of intermittent fasting or caloric restriction. One beautiful example that he has is even when you have triple transgenic mice, animals that they overexpress all the three proteins that they are, seems to be responsible for some of the models of Alzheimer's disease, you can see the response. This is a test, learning test in a water maze. So you put an animal in a water maze and they have to, first you teach them where the platform is, and they need to remember where it is using visual cues. This is what the learning trajectory is over time. And you can see what the triple transgenic does, and as soon as you have them either in calorie restriction or a control animals that is not transgenic or calorie restriction, you can see the improvement both in the speed of learning and also in the retention of that memory. And this is both in males and females. So in summary, what I wanted to say is that in lifespan of most species can be extended by caloric restriction. We know that now there is a lot of questions about the universality of this statement. We know that the discovery of caloric restriction has led to the identification of several molecular pathways which have in turn led to changes in extension of lifespan when you mutate some of these proteins. So when you do genetic interventions that alter the pathways that were raised with caloric restriction, you can affect lifespan. This identification then went into creating a search in molecules that can target those pathways. And we have now done, I think that we are up to 15 molecules that can extend lifespan. When I started doing research in 2000 in aging, there was only one intervention that extended lifespan, and that was caloric restriction, period. So there was no hope to do any kind of interventions. And then I think that there are a growing number of CR strategies that seem to recapitulate the original observation of CR, intermittent fasting, time-restricted feeding, and fasting mimicking diets. So I think that if we keep studying and elucidating the mechanism of CR, I think that we will have a much better understanding of the biological processes behind pure aging. I think that we will have a better success at doing the translation into human health. We'll have a better understanding of how multiple chronic diseases, including the etiology, risk factor, onset, and progression. And I think that we'll have an ability to address the most common presentation of the disease in the population, that they are the comorbidities. Very quick, all these changes in lifespan and with some of these interventions have also been translated into a specific health span. So for example, rapamycin is able to stop skin tumors, induce skin tumors, as well as it does the deposition of beta amyloid plaques in triple transgenic mice. And importantly, some of these investigations has been translated now into humans. And for example, the concept that in the elderly you could reactivate with mTOR, targeting mTOR, the immune system, has led to this paper by the group in Novartis that they gave elderly a rapalog, analog of rapamycin, and they actually improved tremendously the response of this elderly population to a vaccination, which we know that the response is decreased as we get old with age. And then Walter Longo has been working really hard in both periodic fasting and fasting mimicking diets, trying to identify how quickly or how long you have to implement caloric restriction to reap some of the beneficial effects. And he's been very successful in doing short-term intervention, 3 to 4 days of fasting mimicking diet of fasting in the protection against the deleterious effect of carcinogenesis. So I think that, where are we now? I think that we are about here. So we are, I think that the prehistoric area where we are starting to design the original tools that they are gonna help us to move the field forward. And I think that we hope to be here very soon, that we're gonna have a much more sophisticated arrangement of tools, that we're gonna be able to identify el when, el how, and to whom to introduce these interventions. And I think in where we're heading is based on all the longitudinal data that we have in humans and some of the longitudinal data that we're collecting in mice. We're trying to develop biomarkers that then we can test in model organisms to see if this can actually be targeted. And then, through interventions, we can then go back to animal models and do the whole entire validation to get validated metrics that then we can test those into humans. I have to thank everybody in the group and everybody that has either funded me or collaborated with me. One thing that we have to be very careful is how the public is reading all this. And this is, the picture that I used at the beginning, it was in 1992. This was in 2010. So this is kind of, we stop aging. Now is the science of living longer. And now is, there is a pill that's going to cure aging in 2016. Thank you very much.
Thank you, Dr. DeCabo. We have very little time, maybe time for one question. I had one quick one. Is anyone looking at combinations of some of the mimetics and the different timings
So there's
dietary
there are
regimens.
people looking at the combination of There's people looking at the combination of calorie restriction and exercise, and then calorie restriction plus rapamycin, metformin, resveratrol. So and also combination of resveratrol, metformin and rapamycin. So there's been some of them. Right, I mean, the issue is, if you look at, one of the frustrations is that when you look at lifespan, you have to wait 3 years to get your answer. So that is the hint at the end that we really need biomarkers. For example, we're working very hard, Luigi Ferrucci and Stefani Studenski, to try to see, for example, a gait, walking speed, is also predictive of mortality in mice. So then we can have sort of like a biomarker where we can take mice, see how they are declining in the walking speed, and then we can do a treatment and see if we want to correct that. So that's one of the things, yes.
Thank you.
Thank you. Thank all of the speakers, the moderators. Let's give everybody a big round of applause at this point. And thank all of you who've hung with us for the entire day, those of you on the webcast, those of you in person. We will be starting again tomorrow at 9 o'clock. And for those of you who want to have on the food forum our speakers, we will be having a reception for our microbiome in the West Coast. Thank you. Again, thank you to everybody.